in the cell. Again, we have learned most of this information in biology, and so we're going to have to just remember some of that stuff as I'm going through. I'm going to go through this fairly quickly. The mitochondria, we called it in biology the powerhouse of the cell. It is where cellular respiration occurs, where we take in nutrients and oxygen, and we produce something called ATP. The mitochondria has a double membrane. The inner membrane is called a cristae, which is folded. Anything that's folded in a cell in the human body increases the surface area so it can fit in a smaller space. And I just mentioned that we produce most of the cell's ATP, which is our way, or our energy source that we're able to use for any sort of activity. And one of the things that's interesting about the mitochondria is that it contains its own DNA and RNA. And so sometimes you'll hear in crime labs or like CSI type shows that they'll talk about mitochondrial DNA, and they can actually get some information from that. It comes from the mother's side, the maternal side of the family, and they're also able to replicate on their own. So this is what the mitochondria looks like. You can see that inner folded membrane called the cristae, and again, it is making the ATP for the human body. Ribosomes are little dots. We used to represent them as little dots in the cell, and that is where protein synthesis is going to be happening, where we're making the proteins needed for all of the different chemical reactions, etc., in the cell. If the ribosomes are just floating around in the cytoplasm, they are considered to be free ribosomes, and they synthesize soluble proteins. If they're membrane-bound ribosomes, that means that they're going to be attached to the endoplasmic reticulum. They synthesize proteins to be incorporated into membranes, like the cell membrane, those integral proteins and peripheral proteins. Endoplasmic reticulum, there are two different types, smooth and rough endoplasmic reticulum. They are going to be continuous with the nuclear membrane, so they're attached to the nuclear membrane, or the nuclear envelope, as you'll hear it sometimes called. And this is what it looks like. The smooth does not have the ribosomes, and the rough does have the ribosomes. Because the rough endoplasmic reticulum has ribosomes, it is going to be involved in endoplasmic reticulum. It's going to be involved in protein synthesis. Smooth endoplasmic reticulum has lots of different functions in the human body or in the cell, and it again does not have the ribosomes attached to it, and so it has a different function in some of the different organs or organ systems. So in the liver, it is going to metabolize lipids, it breaks down glycogen, and it helps detoxify drugs. In the testes, it synthesizes steroids. In the intestinal cells, it helps to absorb and synthesize and transport fats. And in the skeletal system, in the skeletal and cardiac system, in the skeletal and cardiovascular system, it helps store and release calcium. The Golgi apparatus is a stacked membrane. It looks kind of like stacked pancakes, and it helps to modify and package proteins. And then it's going to form a vesicle to move these different proteins around the cell. And this is what the Golgi apparatus looks like, and you can see that they're budding off into their vesicles to transport certain things. Lysosomes are hold a lot of enzymes. They help to digest things. So in cells where we need to bring things in and digest them, like bacteria, viruses, etc., they are going to really help in that, break that down, and neutralize that so that those cells do not die. It also helps in degrading any worn out cell parts. Peroxisomes is a new organelle that you guys have not heard of from biology. Again, it has enzymes just like the lysosome, and it helps detoxify harmful and toxic substances. So you'll find a lot of peroxisomes in the liver. The nucleus, I think we all remember what the nucleus is. We call it the brain of the cell. It has the nuclear envelope on the outside. It has the nucleolus on the inside, and chromatin, which condenses into chromosomes. And this is where we are going to be going through a process called transcription and translation. So we can make the DNA, we create messenger RNA from the DNA, and then the messenger RNA goes out to the ribosome to make the proteins.